The replication fork is the location in which DNA replication takes place. It is here in which the double helix structure of DNA is opened up to isolate the two strands. Phosphodiester bonds connect the sugar phosphate backbone of the DNA strands. The 3' hydroxyl group end of one nucleotide binds to the 5' phosphate group of another nucleotide. The origin of replication is the location where replication begins. The replication then continues on in either direction away from the origin towards each fork along the DNA strands. Helicase is the enzyme responsible for unwinding the DNA double helix. It does so by breaking the hydrogen bonds between complementary bases. You can think of helicase as a zipper that unzips the DNA double helix as it moves along. DNA helicase unzips the DNA double helix, but something is needed to hold the strands apart while they are being replicated. This is where single-strand binding proteins come in. They attach to each newly separated DNA strand and ensure that they stay separated. The unwinding of the double helix can create supercoils, which can lead to torsional stress along the strands, but gyrase comes in to combat this. Like other enzymes, it helps ensure that replication occurs smoothly. Another enzyme important to replication is RNA primase. It helps to initiate replication by laying down short RNA sequences called primers. These primers give the needed free 3' end for DNA polymerase to begin synthesizing nucleotides. DNA polymerase is the enzyme that synthesizes new DNA. There are two types, DNA polymerase 1 and 3. DNA polymerase 3 is the one that synthesizes the new DNA, whereas DNA polymerase 1 works to remove the RNA primers needed by the DNA polymerase 3 and replace them with the necessary nucleotides. New DNA is synthesized along each isolated strand of the double helix. However, the strands are anti-parallel, meaning one strand runs in the 5' to 3' direction, known as the leading strand, and the other in the 3' to 5' direction, known as the lagging strand. DNA polymerase works in the 5' to 3' direction, which is the direction in which the leading strand is formed without interruption. The opposite strand is formed in the 3' to 5' direction discontinuously. In this case, small fragments of DNA, known as Okazaki fragments, are created and later joined together. Lastly, the enzyme known as ligase seals the nicks between the DNA added by DNA polymerase 1 and the DNA added by DNA polymerase 3. It simply closes the gap between the two. It does not add extra nucleotides to the strand. After this occurs, we now have two new complete strands of synthesized DNA.